PROMICON's project, AIMEN is involved in the development of a spectral sensor which is able to characterize microbiome cultures in the UPC. To do that, we have to perform test campaigns of the microbiome in a photobioreactor.
The first step consists of measuring the total amount of biomass by volatile suspended solids analysis during the growth phase of the microbiome in a photobioreactor. These photosynthetic microbiomes were selected in advance by the UPC. To do this measurement, 3 samples a day are taken. Then we undertake laboratory analysis and high spectral imaging of these samples. Moreover, to know the ratio between the cyanobacteria and other microorganisms, as green algae, each sample is analyzed in the microscope.
Secondly, in order to determine the biomass content, we use a commercial hyperspectral camera to characterize the microbiomes during their growing stage. The sample is placed under the moving platform and then the measurement starts. As the sample is scanned by the camera, the spectral information of the reflected visible light is recorded line by line, generating a so-called hypercube. Throughout this data, reconstructed image is obtained, where every pixel is associated with the spectral reflectance of that area. The spectral data stored in the hypercubes is pre-processed to decide the wavelength bands that provide more information on the evolution of the process, biomass volume, pigment content, and so on.
Finally, to estimate the total amount of biomass from these perispectral images, we do the following. Firstly, we need to pre-process all the data to keep only the information of interest and to remove outliers. Then, we follow a 3-step methodology. The first step is to check if there is any relation between the images and the biomass information from the lab analysis. After checking this, 2 approaches to estimate the biomass from the images are done. For one approach, we use the features extracted from the previous step to train a machine learning algorithm. The other approach uses the images to train another machine learning algorithm, a convolutional neural network. The resultant trained model can be used to estimate a value of biomass directly from the spectral image. The results obtained with both approaches are optimistic. Nevertheless, few experiments have been used. Therefore, the next steps will require the addition of new experiments to validate and to optimize the approaches.